Hi everybody, my name's Dr. Hannah Siddle and I'm from the School of Biological Sciences at the University of Southampton, and I'd like to welcome you to our webinar today. First up, I'd just like to apologise for the technical issues that we had last week, and so we weren't able to go ahead with the webinar. I really apologise for that, and I'm so glad that so many of you have been able to make it back with us this week, which is really fantastic. So today we're going to share with you a little bit about some of the amazing research that goes on in our school, and hopefully to inspire you to study biological sciences as well. So we have two really amazingly talented researchers who are going to be talking to you guys today. So that's Dr. Neil Gosling, who's a paleobiologist, and he's going to be talking to you about some of his research into the fossil record and dinosaurs and some amazing stuff like that. And also Professor Amrit Muda, who's also our admissions tutor and does some incredible research on neuroscience and Alzheimer's disease. Before we pass over to Neil and Amrit, what I'd like to give you is just a bit of, I guess, an overview of the sort of research that we do in the School of Biological Sciences, because in our school, our teaching of our undergraduate students is really led by our research, and hopefully you can get a bit of an insight into that today, and hopefully answer the question a little bit of why become a biologist? Why is this such an amazing area to study? Why it can lead you to so many different careers, and hopefully you get an idea of the sorts of places that this sort of a degree can take you. Okay, so this slide sums up a little bit about the research in a very small way that goes on in the School of Biological Sciences. So as I mentioned today, Amrit Muda is going to be talking to us about neuroscience. So we have a real strength in neuroscience in our school and understanding how the brain works. And I've put in a picture down here as well of some DNA, because we do a lot of work on research on the genetics underpinning different diseases as well. We also have researchers who are working in different areas of conservation, and I always put these little guys up here, and I'm not sure if you know what they are, but they're called Tasmanian Devils, and this is the research that my lab does. And unfortunately, Tasmanian Devils have a really horrible disease, just like humans do at the moment with COVID, but theirs is actually a contagious cancer, and my lab's research is looking at how something like a contagious cancer, so this is an infectious cancer, can actually emerge and spread, and what we can learn about cancers more generally as well. I've also got down here a picture of a plant, because we have researchers looking at crops and figuring out ways that we can increase food production, which is such an important issue in the world today. And finally, I've got an image here that actually possibly could be the images that you see of SARS-CoV-2, or commonly known as COVID-19 in the media at the moment. And obviously that is a huge issue that's facing the entire planet. And we have some researchers in our school who are working on this as well. So I think you can see here that our world is facing some huge challenges, you know, from climate change to disease to feeding the world to conservation. And biological sciences can, the research that we do touches on many of these different themes and can contribute to this. So today I'm just going to talk a little bit about a couple of these different areas, and I'm going to go through pretty quickly because it's just to give you a bit of an overview and an idea of what we do. So one of the big areas of research that we do is actually called biofilms, and it's to do with antimicrobial resistance of bacteria. So you may or may not know that bacteria, when they grow, very often they form these colonies, which, and this is just a little image up the top of the slide, sort of illustrating how this works. And these colonies actually protect the bacteria really well. And they're one of the reasons that we have problems with antibiotics not working and antimicrobial resistance. So we have quite a big research group in our school who are working on ways to break down biofilms more effectively and solve this problem. As I mentioned before, we also have labs working on COVID-19, and this is to illustrate particularly one of the research groups in our school, which is looking for finding better vaccine targets and improving the vaccine targets against coronavirus. This is two of our PhD students who are in the lab working on this. And over on the right here, I'm not sure if you're aware that the COVID-19 virus has what we call a spike protein on its surface. Sorry, and that's how it actually infects and engages with our cells and then infects those cells. This is the spike protein. So when they're talking on the news about the different vaccines that are being developed, these are actually being developed against this spike protein because it's kind of a weak point of the virus. But viruses are clever, and they use a process called glycosylation, which is actually just having a whole lot of sugars around the outside of the spike protein, and they use this to kind of protect themselves and to stop the immune system from being able to see this spike protein effectively. So some of the researchers in our school are figuring out how we can better understand this process of glycosylation, or the addition of sugars, and how we can use this then to be able to make better vaccine targets. So that's some really exciting research that's going on at the moment. We have a lot of researchers looking at climate change, and there's collaborations all over the university, but particularly in geography and environmental sciences, to understand about how our climate is warming and the impact that this is having on our ecosystems, and that includes, of course, flora and fauna as well. And we also have a lot of researchers working on wildlife conservation. We have some fantastic zoologists within our school, and we do research on all sorts of different animals and conserving these in their native wildlife. And just over on the right here, this is a little video of a little Tasmanian devil who was in this trap, and one of my PhD students was out in Tasmania trapping these guys to have a look at these infectious tumours that they have at the moment, and this is one of the ways that we then capture them. And of course the devils, once they then get out of the trap, are really happy to see the back of us, and they run away pretty much as fast as they can, except for occasionally when they get so scared when they get let out of the trap that they actually just freeze, and they just stand there for quite a while until the researchers gradually move back and away from them. But they're a really interesting species and disease to look at as well. And finally, we have some fantastic evolutionary biologists, and you're going to hear from one of those in just a moment, Dr. Neil Gosling, and you can just see him in his very handsome hat there, standing there underneath the statue. And this is actually, he runs a really fascinating master's course on evolution, and every year, when it's not a COVID year, obviously, he takes his students to the Galapagos Islands, and they do some fantastic research projects out there. It's an amazing experience, and I'm sure he'll tell you a little bit more about that in a moment with some of his research. And as I said, that's just a bit of a taster of the research that goes on in our school. And we are a research-led university, which means that kind of underpins all of our teaching of our undergraduate students. And so this is just to give you an idea of what our building looks like. And so this is our quite new life sciences building, so that we've only been in for about 5 or 6 years, I think. And this is just the steps that are going down to our teaching labs that are in the bottom of this building. So this is where all of our practical classes are held, tutorials and so on, when our students are on campus. And just to give you an idea, this is some of our undergraduate students who are hard at work in the labs at the moment in these images, and this gives you a little bit of an idea of what our teaching lab spaces are like, because all of our degrees are practical based degrees, and so they do all have a practical component to them. And finally, this type of degree, so a bioscience degree, can take you so many different places in your careers. So within our school, we have degrees in biology, zoology, biochemistry, biomedical sciences, pharmacology and neuroscience. but we find that our graduates go on to all different sorts of areas. So we have some that go into either training them to be a doctor and medical sciences, or more into clinical science. We have graduates that go then into research and analytical sciences. We have some that go into agriculture and environmental sciences, and then others in maybe careers that you may not have thought too much about, like forensics, patent law, pharmaceuticals, and so on, and also, of course, teaching as well. So this is some of the places that these degrees can take you. So what do you need to do to become a biologist? Well, I'd say the main thing that you need is curiosity about the world around you. But there's also a really great array of degrees in biological sciences. And we'll be really happy to take any questions that you have that you can post in the chat of the webinar today. So if you've got any questions at all, there's no need to wait till the end. You can just pop those in the chat, and we'll be able to get back to you as the webinar goes on, or if there's anything that we don't get to, we can address it at the end of the webinar as well. So please don't be shy with the questions. We're really happy to answer anything that you want to ask. It could be about the research, it could be about studying, it could be about the type of degrees that are out there available for you to look at. I am aware that your Ucas deadlines have been delayed a little bit, so if we can help with answering any of those questions, we're really, really happy to do that. So without further ado, I'm going to hand over to my colleague, Dr. Neil Gosling. And I think this image here is a really apt one for Neil, because he's going to tell you all about some of his research into dinosaurs and the fossil record and evolution. And I think you're going to really, really enjoy it. So thank you very much. And our team will now switch over to Dr. Gosling. Thanks, Neil.
Thank you, Hannah. Okay, so hopefully you can see the title page of my talk. So I'm going to talk to you about the history of life on Earth, and I'm going to take you through an awfully long period of time in about 20 minutes. Okay, so thank you very much for coming to listen to me, and I will start now. So I'm a paleontologist, and I am interested in understanding the way that life has got to be the way that it is today. And also you can use historical events, you can use extinction events, you can use the changing climates throughout the past to help us get an idea of what's going to go on in the future. Okay, just trying to get that to advance. It's decided, there we are. So geological time is a really hard thing to get your head around. The Earth formed 4.56 billion years ago. The first cells formed between 3 and 4 billion years ago, and insects appeared 500 million years ago. So even the shortest period of time here, 500 million years, the time is just, it's something really difficult for us to fathom. So one of the things, geological time is, we human beings appear at this point here, but this beautiful diagram, and I really like this diagram, doesn't give you an idea really of the scale of life. So maybe we can turn it into a clock. We understand clocks. So if we have a face of a clock, we get the origin of the solar system starting here, and it moves its way around. And humans again appear in the very last second of the very last day, of the very last minute, rather, of the last hour in the 24-hour cycle. We can't get the time. You know, it just doesn't work. There's too much of it. It's just too much time. Okay, so can we explain time in a better way than using a clock? And I think we can, because humans are really good at understanding spatial distance and getting good perception of these ideas, rather than simply it's to give us, because time is a very abstract concept, and if we can apply it to something, to a journey, for example, and we can put time points along that journey, well, that starts to make sense a little bit more. So I like to start my journey as an evolutionary biologist, and being someone who's read The Origin of Species a number of times, which was written in 1859. I think that all good journeys in history of life should start at Big Ben, because Big Ben bonged for the first time in 1859. So what we're going to do is we're going to take a journey from Big Ben all the way through along 125 kilometers, all the way down to my desk in Southampton in the present day. So Big Ben, it represents the formation of the Earth. My office, where I'm sitting now, represents the present day. And we can start to ask questions as we move through here about what events are happening as we're going along our journey. And ultimately, we can ask our question, how far from my desk did humans diverge from chimpanzees? Chimpanzees are our closest living relative. So we can start our journey. Here we go. And at 3.8 billion years, probably more than, actually, probably closer to 4.1 billion years, life is absolutely rock solid, preserved in the fossil record, in the form of these structures called stromatolites. And Hannah alluded to biofilms, these sheets of bacterial film. Well, actually, stromatolites, each one of these lines represents a biofilm, which then gets covered up with sediment, and then another biofilm forms on top, and so on and so on and so on. So you have eventually these huge domed mushroom-like structures, mushroom-shaped structures of biofilms building up on top of biofilms on top of biofilms. And these represent our oldest fossil record. We have to come all the way along to just north of Basingstoke at 2.1 billion years for photosynthesis to happen. If anyone's from Basingstoke, I'm not casting aspersions or anything like that now, but that's where eukaryotic cells form. And we've come already 2.56 billion years before we've even got cells with a nucleus. So nothing particularly, well, it's hugely exciting in terms of the origins of life, but there's no big organisms. There's nothing. This is all single-celled organisms which are still sitting, doing their thing. But we've already come through more than half of the age of the Earth. We come down to just north of Winchester. We are now 900 million years ago before we have evidence of sponges, animals. Okay, we are now. That's within about 20 miles. It's not even that far. It's 15 miles from my desk. Okay, about 25 kilometers. So now we're coming along from Winchester. Winchester is about 9 or 10 miles, and this is where we end up with bilateral symmetry, the first evidence. And what we know is that we have little trackways that can only have been left by something that has a bilaterally symmetrical body with its legs moving either side of its body. And this is happening in Winchester. This is about 15 kilometers from my desk. We come south, and this is where the Cambrian Explosion takes place at 535 million years. And by this point, we've come 8/9 of the entire history of the age of the Earth has happened before we get to Winchester, before we get to the Cambrian Explosion. And we are now less than 9 miles from my desk. We come down now, we're just near the golf course on the M3, Otterbourne Golf Course. It's the next turning. We've got land plants at 456 million years. Now we move down, and we're at 416. We have the first fossilized insects. At 397, we have the first evidence of tetrapods, so animals with 4 legs, and we are now less than 10 kilometers from my desk. So all of the history of life so far has brought us to within 10 kilometers, okay, of my desk. And this is the first point where we end up with animals with 4 limbs. We come down to Chandler's Ford. If you come to Southampton, you'll notice that in Chandler's Ford, there is a really huge Asda's. Lots of students go there. And that's where the dinosaurs appear in the fossil record. And 96% of the time since of the age of the Earth, 96% of the whole age of the Earth has passed by the time we get to the dinosaurs, okay? It's a huge amount of time, and evolution has generated these wonderful forms, but these icons of the fossil record, they only appear within the last 9th of the age of the Earth. We don't get birds until we come to the turning for the M3, almost joining with the M27. We are now within about 5 kilometers of my desk, and then we get flowers as well. And now we're into really recent history. If you were to come to Glen Hall, if you were to stay at one of the halls, it is less than 1.5 kilometers from my desk. Glen Halls is where 66 million years ago the Cretaceous Tertiary extinction event happened, which wiped out the non-avian dinosaurs and the pterosaurs and all of the other exciting organisms that we are familiar with. And 98.5% of the time since the Earth has formed has passed. We're now at the junction of Burgess Road and Hill Lane. This is the point where monkeys appear. We are now 0.5 kilometer from my office. We're now right on the concourse. 99.87% of the time since Earth has formed has passed. We are on the concourse outside the building, okay? 6 million years ago, just at this point here, this is University Road, my office is here. We are now about 45-50 meters from my desk, and humans diverge from chimps. We come right onto the concourse. There's a lovely flagpole. This is where Ardipithecus, a fairly human-like skeleton, 4.5 million years ago, that's where that diverges from us. Homo erectus is just outside the back door by the coffee shop of our building. So in very, very recent history now, anatomically human skeletons appear 5 meters from me. There we go. That's 1 meter. So 5 meters from me, just along from my office, that is where anatomically human skeletons appear in the fossil record. This far from my desk, from where I'm sitting now, on this scale, 81 centimeters, Neanderthals die out. 27 centimeters is the last ice age. Agriculture is at 24 centimeters. Where's it gone? I've lost my 24. There we are. Biblical creation as described in the Bible, 15 centimeters from my desk. Okay. And your birth, I had to go and pinch one of my wife's darning pins. Your birth, or rather, in fact, really, a whole human lifespan on this scale is less than the width of this darning needle. So why is this important? Why is getting an idea about geological time so important? And the main reason for this, I think, is because given the enormous amount of time that we have, rare events can happen. One of the arguments against evolution, which is thrown up, is it's just the chances of these things happening. It's so unlikely. However, when you've got the huge expanse of time that we have, okay, the chance of a mutation happening that is going to be beneficial is very likely. Okay, there is a 1 in 100 million chance of any DNA base pair mutating in any given generation. But you've got 3 billion base pairs, okay, in a human genome, which means that 30 base pairs will mutate. So in a population of 250,000 humans, there will be 7.5 million mutations every single generation, which means that humans and chimps that split 6 million years ago, just about 45 meters from my office, okay, if the generation is 25 years, every single DNA base can have mutated 240 times. And this means that the adaptive landscape, your genetic makeup, can have been well explored. Now, most of those mutations won't be useful. They won't be beneficial, but every single one can have mutated 240 times, which means that everything that can have happened, and all of these rare events, they can have happened. So, the history of evolutionary theory. I just want to give you a little idea about how we've come up with the ideas that we have. So, in the same year that Darwin was born, a Frenchman by the name of Jean-Baptiste Leconte de Lamarck wrote his book, Zoological Philosophy, and that was the first idea written down that gave an idea about evolution, about changing species. Darwin published The Origin of Species in the same year that Big Ben bonged. Gregor Mendel, a monk in the Czech Republic, talked about peas and genes and showed that there was the mechanism of inheritance, but unfortunately his work was lost until 1900. But then his work was rediscovered. Haldane made observations on the peppered moth in 1920. People were starting to think about genetics and how that could fit in with natural selection. People were doing inbreeding experiments to actually understand how genes interacted with one another. Dobzhansky wrote Genetics and the Origin of Species. We have Systematics and the Origin of Species. And then we pull all of these things together: morphology, botany, cytology, so cell science, genetics, paleontology and fossils, hooray! Ecology, systematics, all of these were pulled together to give us the fusion, the modern synthesis in 1942. And this was the reconciliation, the complete reconciliation of Mendelian genetics with natural selection. So macroevolution on a geological scale, so the changes in species, the evolution of dinosaurs, the appearance of birds from dinosaurs, exactly, and microevolutions in existing populations can be accounted for in exactly the same way. Microevolution is just a little tiny bit which, building up, gives you macroevolution. So to sum up, you need to have heritable variation, you need to have natural selection, and you need to have a huge amount of geological time, and that is enough to give you evolution by natural selection. And the reason I'm talking about this is because no matter what biological science you study, evolution is the mechanism, the underlying mechanism for all of biological sciences, okay? So no matter what you do, having an understanding of how species form, how genes interact with one another, and how they change, and how they understanding comparative anatomy, and all of these aspects that feed into evolution is fundamental to all biological sciences. And you might ask yourself, well, okay, that's fine. I want to go off and I want to study genetics. I want to go and do cancer biology, and that's wonderful. But actually, understanding zoology, understanding ecology, understanding biology, the less applied versions of biological sciences, is just as important. And one of the things that I run at the moment is a Facebook page, because understanding evolution, understanding zoology, being interested in this, zoology is really important. Because one of the things that, if anything we've learned at the moment, is that connecting with nature again, understanding the world around us, is really important to help us come back together again and get through this time of COVID. So thank you very much for listening to me, and I hope if you've got any questions, I'll be able to answer them in a little while. Thank you.
Okay, so I guess that's me. Can you guys hear me all right? Okay, great. I'm Amrit Mudar, and I'm a professor in neurosciences, and I'm also the admissions tutor. Our idea really for this webinar series was to give you an indication of what a degree in biological sciences really means and where it can take you in the future. And in this current situation, the pandemic situation that we find ourselves in, it's very clear that scientists make a very big difference. All the scientists around us are working very hard to find out ways in which the virus, the COVID virus, works to cause the detrimental effects that it's causing, and how vaccinations can help and how other methods can help. This is just one example that's come to light at the now, but there are many, many, many different ways in which biomedical scientists, neuroscientists, biological scientists can make a difference. And we chose two different talks to give you because we wanted to make sure that you appreciated not just where a degree in biological sciences can take you, what you can do with a degree in biological sciences. I say biological sciences, but really what I mean by that is a degree within biological sciences. We've got a number of different programs in biological sciences, as you've heard already from Hannah Siddle. But I also wanted to emphasize to you how the teaching in Southampton is very much research-led. We are a Russell Group university, which means that you are taught by individuals who are actively engaged in carrying out research in their area of research. I'm a neuroscientist. My area of research is diseases of the brain, such as Alzheimer's disease, and much of the lectures that, many of the lectures that I give relate to neurosciences, and in fact, in the third year, I give lectures in the second part of the year, semester 2 lectures, which will be starting in a couple of weeks, where the module that I coordinate is all about diseases of the brain. And in that module, I will talk about Alzheimer's disease, but I will be giving students information that's very current, that's very relevant. I don't need to go and read up about it. I'm telling them what's the latest in this area of research. And that's what you get if you come to a Russell Group university. In fact, I don't know whether other Russell Group universities operate in the same way, but in Southampton, every member of staff who is working on an aspect of research then teaches on modules so that the students get the most current information. Okay, so I'm going to take you through a very quick whirlwind tour of the research that I do, and it gives you an example of the type of thing that a scientist can do. And this is what I would teach in the final year. And if students came to work in my lab, at the moment I've got 4 students working in my lab, they would work on an aspect of this project, of a project such as this one. So I work on fruit flies, but before I kind of, before you start sort of thinking, what can fruit flies tell you about Alzheimer's disease? How is it relevant? Let me first just remind everybody what Alzheimer's disease is. Alzheimer's disease is a condition in which there's a lot of cell death in the brain. So you can see in the middle panel on the top, you see the brain of an individual who had Alzheimer's disease before they died. You see a lot of shrunken, gaping holes inside on the surface of their brain, which is effectively telling you a lot of cell death has occurred. For comparison, this here at the bottom is the brain of an individual who died at an equivalent age, but this a very nice plump looking brain with no evidence of cell death. I bet you the person on the bottom who had the brain on the bottom really had a bit problem with their day-to-day functions, with their memory, whereas the person on the top had a lot of learning and memory difficulties. If you look inside the brain of someone who has Alzheimer's disease, as was done by Professor Alois Alzheimer, who first described this disease in the early 1900s, and he described it because he looked at this very patient here on the right-hand side. Auguste D is the name of the patient. She had a very profound memory loss, and she was a housewife, but her memory loss was so significant that she couldn't carry on with the day-to-day running of her house. And when it became very serious, So we're not just talking about forgetfulness, we're talking about actually being unable to do anything that makes you unsafe amongst other people. So in today's, to give you an example, in today's day, in today's times, someone who has Alzheimer's disease perhaps would park their car in a car park, would then go to the supermarket or wherever else they go. They would never find their car when they come back, because the memory loss they have is so severe that it interferes with them then being able to conduct day-to-day activities. So for them, they would not be able to find their car to drive home. They'd have to be driven home by someone else. Normal forgetfulness is just not knowing where your car is, and then after a little while you kind of find it. People like me drive bright orange cars so that I can spot it in the car park. But that kind of gives you an indication of what I mean when I say memory loss that severely impacts your ability to carry on with day-to-day activities. But coming back to the brain, Auguste was looked after by Professor Alois Alzheimer. When she died, he looked inside her brain and he described for the first time two structures that I've got pictures of here. One called a plaque, which is on the top, one called a tangle, which is at the bottom. It doesn't matter the details of what these structures are. It's just to tell you that this is not just age-old forgetfulness. This is the appearance of some abnormal structures in the brains of people who then suffer from this disease. And there are many questions that a scientist would ask if somebody knew that in the brain of someone like Auguste you had these plaques there and these tangles. What would the scientist ask? What would the questions the scientist would ask? First of all, the scientist would want to know, what is this abnormal structure made of? That'd be the first question. These abnormal tangles that we see at the bottom. So this here is the outline of a nerve cell, and that dark structure there, that flame-shaped structure, is a rope-like structure that grows inside those nerve cells. It squeezes the life out of them. Those are made up of a protein called tau, because that's what a scientist would first ask. What makes up this tangle? The scientist would also ask, what makes up this plaque? If you were sitting right in front of me and I was giving you this lecture in my lessons in Southampton, or if you'd even come to a physical visit day, I would pause and I would ask you, if you were the scientist, what would be the next question you would ask? But because I can't do that, I'll just have to ask you, I'll have to just tell you what I think the next question would be. And the questions that my students in my lab ask are, okay, we know then that this normal protein called A beta, this normal protein called tau, that's found in all our brains, somehow they become abnormal to form the tangles and the plaques. How and why do they become abnormal to form the plaques? How and why do they kill the nerve cells within which they form? And how does that then make somebody lose their memory? Another question my students would ask is, if these are normal proteins that are forming inside these nerve cells, they are normal, but they are found in the nerve cells in all our brains. When they become abnormal, how do they make those nerve cells sick? How do they kill those nerve cells? And how does that lead to memory loss? And these are the kinds of questions that a scientist can ask. And in order to answer these questions, one needs to recreate this process of the tangle formation or the plaque formation and study the sequence of events from when they formed to what happens to give you the memory loss. And of course, you can't do those studies in people, so you employ model organisms. And I have chosen to do this work in fruit flies. Fruit flies are those humble flies that just hover around your fruit basket, particularly your bananas when you've forgotten to eat them. They have been the geneticist, the biomedical scientist's friend for decades. Many people have won Nobel Prizes for the sorts of work they've done with fruit flies. I don't have time to tell you about those studies, but what I can tell you is they are very useful within which to recreate these pathologies to effectively. The work I'm going to tell you is I recreated in my lab with my students, we recreated these abnormal tau structures inside the brains of fruit flies to study how they make nerve cells sick. And we were able to do that because we know that many of the proteins that are found inside human brains, in humans, have homologs. We call them homologs, but what we mean by that is there are equivalent proteins in flies. So a lot of what you study in the fly will be relevant to people, because the protein in the fly that does a particular job, its equivalent protein will be doing the same sort of job in the in people. So if you can make this tau protein abnormal in the fly brain and see what it does, it's very likely that it's doing the same sort of thing in people. The fact that flies are very cheap to do experiments is a great help. If I wanted to do the same experiment in a mouse, it would cost me 100 times more. The short lifespan of the fly means that a disease like Alzheimer's disease, which occurs over the lifespan of an individual, I'd have to study a human for 70 years to study the disease. If I did this experiment in a monkey, I'd have to study the monkey for 15 to 30 years. If I did the experiment in a rodent, I'd need to study the rodent for 2 years. In a fly, I can get that answer within 2 months. So it's a lot quicker. And of course, I have some very elegant tools. I can create some very nice fluorescent markers of cell death, of neuronal function. And this is an example here that I'm going to give you. But first of all, what was I asking? I was asking, how does an abnormal tau protein, this is the protein, this protein tau, it's what forms these rope-like structures in nerve cells. My question with my students was, how does that make nerve cells sick? When it becomes abnormal, how does it make nerve cells sick? But to answer that question, I needed to first understand, what does tau normally do in nerve cells? And so for that, very quickly, you look at a nerve. A nerve has a cell body, which is its housekeeping hub. It's where a lot of its activities take place. It's got a connecting process called an axon. An axon is really a highway where material that's made in the cell body is transported. And the synapse is its business end. So many of you that are doing A-level biology will know the synapse is where neurotransmitters are released. It's the point at which the connections are made. Memories are really consolidated and generated even at the business end here. So if you look in the axon, tau is found there normally. In this axon, there are many tracks that allow cargo, which contains vital materials like neurotransmitters. They're made in the cell body and they get transported down these tracks to the synapse. Tau proteins are normally found on these tracks. These tracks are very unstable. They break down all the time. They need to be held in place with connections, really connectors, sellotape type proteins, and tau is one such protein. Again, if you were sitting right in front of me, and if you were in my lecture, if you come to the university and you come to the third year, you must, raise your hand and tell me, Dr. Mudda, I listened to your lecture and you were meant to ask me this question. I will answer it now. The question I would ask my students is, if the tau is abnormal now, like it is in Alzheimer's disease, what do you predict will happen? Okay. So the first thing is you'd predict that that ability to hold this track together actually should become affected, because normally this protein holds the track together. If it's abnormal like it is in Alzheimer's disease and it's now not worried about holding the track together, but it's going away clumping to itself and forming rope-like structures, what happens to these tracks? And then what happens to transport? So what we did was we created some very nice transgenic flies. This is an example of the elegant tools we have. And our transgenic flies effectively had a fluorescent protein, a green fluorescent protein in the cargo, the vesicles that go up and down this highway. So this highway here, and you have the cargo that goes up and down on the tracks. We put a green fluorescent protein in there because we can with the flies. And you could see that in control animals, there's lots and lots of cargo all the way up and down. We did this in animals that we anesthetized, and we looked in real time through their nerves. Again, something you can do with the flies. And we were able to visualize this movement of cargo up and down the tracks. It was beautiful, like you're eavesdropping onto a city at work. But when we now express the abnormal tau, exactly as we predicted, this transport of cargo became very severely disrupted, because the tau should be in there. It should really be keeping those tracks together. We don't know what it's doing because it's abnormal. And you see these traffic jams now. The cargo really isn't getting to the end. This nerve is still alive, but the cargo isn't getting to the end. Why does this cargo not get to the end? Why does the presence of the abnormal tau stop the cargo getting to the end? And again, if you were sitting in front of me, I'd ask you to predict. My students would do that. So normally this is what you think would happen. But if the tau becomes abnormal, we think that it just falls off those tracks, and those tracks need the tau to keep it stable. And so when the tau falls off the tracks just fall apart. And here are real experimental data that we published. There were students on this paper who contributed to this work, who were then authors on this paper. So if you look at the tracks, if this track was effectively running out of the screen because the nerve was coming out of the screen, if you slice it in the middle, you see these cylindrical structures in normal animals that tell you the tracts are intact. If you then look through the cross section of a nerve where the tau is abnormal, you don't really see these cylindrical structures. The tau's really fallen apart exactly, or rather the tracks have fallen apart exactly as we would predict. So this hypothesis, this is exactly what the scientist does. You predict, you hypothesize a mechanism by which the nerve cell is getting sick, and then you predict that this may be how and why the nerve circuits in the brains of people who have Alzheimer's disease, who have these abnormal tau structures, is probably now starting to become that's how the nerve cell is starting to become sick. Again, if you were sitting in front of me, I would ask you, okay, it's all well and good knowing this is how the nerve cell is getting sick. How can you use that for therapy? How can you now say, I know if I look at an Alzheimer's patient, inside their brains, transport isn't occurring because that tau isn't binding to the microtubules, they're falling apart. How can I use that information now to devise a therapy? Again, that's what the scientist does. So my students and I, we predicted that if you now use drugs that sellotape the tracks together, there are such drugs. Chemotherapy, for example, is a mode of action of chemotherapy drugs is they sellotape together microtubules, which in the case of cancer cells is a very good thing for cancer cells because it makes them very stiff, the microtubules very stiff, and stops them from dividing. In this case, we wanted to stiffen broken down microtubules. So we fed these flies a drug, paclitaxel was the name of the drug, that effectively put those microtubules together, and we predicted that the tracks would get sealed together, stapled together, sellotaped together, and axonal transport would resume. And indeed, this again is an image from real experiments that we did. This was a subsequent paper we published 10 years later. It takes this long to do. What I've told you in 10 minutes took us 10 years to do, led to 2 very good papers. But again, you see here the tracks are broken down in the animals that had abnormal tau. But look, this is the sibling of this animal, right? Born off the same parents, but where this one wasn't given the drug, this one was given the drug. Within 24 hours of getting the drug, look, there are so many small microtubules. And this is a very good example of how knowing what the biomedical scientist does, understanding disease, this is different to what a clinician does, a physician. I started my career thinking I wanted to become a doctor. I started medical school. I left after a month because I realized that I didn't want to diagnose illness. I wanted to work out how the illness appears so that I could work out how to stop it. This is exactly what I'm doing. I'm working out how the illness appears so I can work out how to stop it. The drugs that we used went into clinical trial and they had beneficial effects on Alzheimer's patients. And I want to stop there. I apologize if I've taken longer than I should, but I really wanted to give you a very quick whirlwind tour of what being a biomedical scientist is about, what you'd expect if you come to Southampton and are taught by people who are engaged in actual research, and how you can contribute to this if you manage to do a project with them. So I'm now going to stop sharing my screen, and I'm going to hand over to Hannah so that she can tell us, so we can continue to answer your questions, but please do ask questions.
Yes, that's right. As Amrit said, please, if you've got any other questions, please do continue to put them in the chat. We've got another 10 minutes to make sure that we've answered everything. And as I said, it can be research related, it can be to do with Neil's talk, it can be to do with Amrit's talk, or it can be to do with our degrees, any of those things. And we're all here now, so that's nice. So yeah, we're really happy to answer anything. Amrit, there was just 2 that came up while you were talking. One was someone asking about studying neuroscience and understanding addiction. Does that help us with understanding addiction?
Yes, absolutely, absolutely. I saw your response then. I was about to type, but it's possibly better for me to say it out aloud. So addiction, neurodegenerative disease, these are all disorders of the brain. It's where the brain, the nerve circuits, the circuits in the brain have started to become dysfunctional. Whether that then leads to addiction, it just means that the circuits that are becoming dysfunctional there are those that are involved in reward. And addiction is where they're activated too much, and then you become addicted to getting that reward. So it is a neuroscience, we have a neuroscience module in the final year where you can study that. So in the final year, 2 examples of the neuroscience modules are, one is the neuropharmacology of CNS disorders, where you would study addiction, you'd study schizophrenia, you'd study depression. And the other module is the one that I run, which is about neurodegenerative diseases, such as Alzheimer's disease, Parkinson's disease, Huntington's disease, where there's degeneration in the brain. So you absolutely can learn about it in this degree, but then you can afterwards go and work. If the question there was, can it enable us to go and work with people who have addictions? Well, I put a link in the chat for clinical training programs. They require you to do a first degree in biological sciences, and you can work there with neurologists who, that could be the clinical training program you go on to do that, if you were interested in that. And of course, you could do a PhD or a master's in a lab that studies addictions. So that was one question. Hannah, what was the other one?
There was another question. I think Neil actually has answered it really well, but it might actually be worth sharing this with everyone. It was just about students, the students that you were referring to, AMRA undergraduates. So I think, Neil, if you want to share that answer.
Yes, so absolutely. We involve students in our research at all kinds of different levels. So personally, I have students who've done their 3rd year projects or their undergraduate master's projects with me. I've also had students who've just come along and said, Neil, dinosaurs are cool, can I work on them? And as soon as people say, what you'll realise is actually that as we become middle-aged and start to feel irrelevant, it's really nice to have young, excited people come and massage our egos by saying that what we're doing is important and interesting. And if you come along and see us and say, I'd like to do something, you know, I'd like to work with you, I'd just like to get involved in your research, then absolutely come and, you know, there's the formal way through research projects in your degree, but actually there's students who are getting involved in our day-to-day stuff as well. And it's not always the most exciting aspects of the research, but it's the real research and it's absolutely vital. And I'm writing up projects, papers at the moment who are undergraduates, and they're going to have their name on papers. So it's phenomenal, and it's how I got involved with starting to do research, and it's how most of us, it's showing that interest, showing that excitement, and coming along and just wanting to get involved. And we'll never turn you away. We'll always find you something. And it's
a lot of our undergraduates as well do summer projects with us as well. Yeah. And or just kind of come and volunteer in the labs, and some of the summer projects we can find internships and scholarships for them. Not always, but sometimes we can. So there's always opportunities to get involved in the research in the lab. Yeah, like Neil said. And actually there was another question there as well that I think we've answered, but it was asking about how many of our master's students go on to do PhDs at Southampton, and we did a rough estimate of 5 to 10 a year, and that's not just in biological sciences though. So some of our students go and do a PhD in medicine or in ocean and earth sciences, and then of course they might go and do PhDs at other universities as well.
Yes.
So
yeah, I think the number's possibly a little bit larger than that, Hannah, only because it's very competitive to get a PhD placement. And so sometimes students don't necessarily go and do a PhD the first year after they graduate. This year, I've been writing lots of references for students who did a master's program with me a year ago, right? They graduated, they then decided to think about which PhD they wanted to apply for. And they'll eventually start it 2 years later. So they don't, for instance, come into our accounting, but I think about 10% is a reasonable estimate for those that go on to do PhDs, but they certainly, I mean, a master's qualification and a PhD, and this sort of relates to a question I have there about what careers can you go into. These are no longer things that mean you are stuck to working in a lab. I love the idea of being stuck to working in a lab, but not everybody does, right? But what it means is we understand that the scientist of today, the neuroscientist, the paleontologist, the biochemist, the scientist of today doesn't necessarily need to apply their knowledge in the lab or in the field. They are very much applying their knowledge when they go and work for a pharmaceutical company, where they go and work for outreach, where they go and work for, some of my PhD students have gone on to become the outreach officers of Alzheimer's Research UK, where they needed to have a PhD in Alzheimer's disease to understand the disease, so that when they go out and raise, engage in events to raise funds, or when they need to tell funders what the money has led to, what research it's led to, they can actually make sure that they're factually correct, because they understand the research that's been done, and they can make sure that they can convey it in a language that is easy for the funders to understand as well. And these are all additional skills you pick up in biological sciences. Over the 3-4 years you're with us, you will learn how to articulate your thoughts. You will learn how to speak to an audience so that they understand what you're trying to tell them, that you're exciting them, you're motivating them. And those skills actually then put you in a good stead wherever you decide to apply your scientific knowledge. You'll all be scientists, okay? A scientist is not just like me, Neil, and Hannah. You're all scientists when you graduate. You'll just be applying your scientific knowledge in a different area, and we'll help you get there. We absolutely will help you get there.
Someone else has just asked if there's any chemistry or pharmacy webinars. I'm afraid this one we've just organised off the back of biological sciences, but if you have a look on the University of Southampton webpage and have a look at the School of Chemistry, there'll be some contact details of people that you can contact about chemistry. And for pharmacology, we do offer a BSc in pharmacology through our through biological sciences as well. Someone else has asked you, how long does it take for Southampton to respond to a Ucas application?
Yes, I can tell you that. And then after that, Hannah, if you can answer Selena's question about a typical first year, I can answer the question about a typical biological sciences turnaround period. So we consider every application, we read all the personal statements, and you can imagine you should get a response from us within 20 working days, because we have a lot of applications and it takes us that sort of time to make sure we've read all the personal statements and we kind of agree that you're the right person for us. And if you're not the right person for us, from your statement, we will interview you. If you are concerned, I will put my email address at the end of this, or it was actually at the end. I can put it, I can ask the producer of the show to put my email address as an announcement. Please email me. If you've put in your application and you haven't heard from us within 20 working days and you're concerned, email me and I will chase it up personally. Okay, Hannah, over to you for Selena's question.
Yeah, so Selena's asking, what would a typical biological sciences first year look like? So actually our first year courses, no matter what degree program you're doing, all look very similar, although there is some differences between them, but there's some elements that are quite similar. So everybody does some physiology, and that's human and animal physiology both. We do some cell biology and some genetics. We do a little bit of biochemistry, a little bit of plant sciences. And so the idea of our first year is to get everybody kind of to the same level, so that when you're going into second year and third year, you're ready to specialise depending on what degree program that you've chosen. And that first year, although there's not any formal research projects like there is in your third year, but there is an important practical element. So nearly all of the modules that you'll take in your first year have practicals in them. Okay, so you'll be in the labs, you're in the labs every week doing your practicals. So you're getting those practical skills so that when you do your research project in your third year, you've got those skills that you can call on. So you do get some research in your first year, but more in your third year. In saying that, there's nothing stopping you from contacting individual academics and lecturers. Yep. If you've had a lecture and you really enjoyed, they might have told you a bit about their research and you really enjoyed it, then you can contact them and see if they've got anything in the lab that you could get involved in.